Good afternoon and welcome back once again to NASA's Kennedy Space Center in Florida, where we're here now to talk about some of the important research and technology that is going to be supporting the International Space Station with tomorrow's launch of SpaceX CRS-5. And I'm pleased to be joined on our panel by Julie Robinson, the International Space Station Program Chief Scientist from NASA's Johnson Space Center in Houston. To Julie's left, Kenneth Shields, Center for the Advancement of Science and Space, or CASIS, Director of Operations and Education. Cheryl Nickerson, Micro PI, Principal Investigator from Arizona State University. And Sam Durrance, NanoRac's Sable Principal Investigator from the Florida Institute of Technology. And we'll begin with presentations, after which we'll be happy to take your questions. Julie?
Thanks a lot, Mike. So, we say that ISS is really off the Earth for the Earth. And one of the things that you'll see in this briefing today is some of the variety of different research that's going on inside the pressurized module of the ISS, usually with astronauts helping out in different ways. It's really hard to come up with a panel for you nowadays compared to years ago, because the number of experiments has grown so much and the number of researchers has grown so much. Right now, active on orbit, there are about 250 investigations. 94 of those are brand new to this 6-month period, and about 432 researchers have their work ongoing on the ISS. And we could bring you only 2 of those researchers today, and we wanted to bring you one of a number of different themes that we could share with you about the research going on, especially the research going up on the SpaceX 5 flight. And so this is just one theme among the different technology demonstration, biology research, research in human health, research in physical science processes, and earth and space science, some of which we talked about at the last briefing. The theme that we chose today is something that's really been changing on board ISS. If you recall, the last space station flight, we had our first rodent flight, and on this next SpaceX flight, we have a number of different biology investigations going that use what we call model organisms. Now, model organisms does not mean, high heels walking down a runway organisms. It really means the way that we use different creatures to understand the basic overall universal principles of biology. So if I could have my graphic, this is just kind of a way of showing this. On the lower left, you see the basic structure of living organisms, the DNA, the proteins, and the cells. And then we have a set of simple organisms that we understand the genetics on very clearly, like simple plants like Arabidopsis, fruit flies, which are also called Drosophila, rodents like mice or rats, and worms, such as this roundworm C. elegans. And we use all of those as information to help us understand how living organisms work in general and to give us insights into how humans work in particular. It may seem amazing, but the genes that are shared between humans and those organisms in the middle are a large majority of the genes that we know about. And so you can use different organisms in different test cases and have large sample sizes, which you're never going to get with astronauts. When we do a study with astronauts as a subject, we might call 12 subjects a good experiment, and that takes us several years to accomplish. But when we do research with model organisms, we can have dozens to hundreds to thousands of organisms to use for the research, and so that gives us a lot of statistical power, and that's one of the other reasons that we use model organisms. So just to give you a sense of some of the model organism experiments that are on this flight, and some of those investigators are here, some of them are not, but NanoRac's Sable, which Sam will talk to you about, is looking at proteins primarily and actually their physical interactions. Triple Lux B is a European Space Agency experiment that is looking at both cells from mussels, the mussels that live in the ocean type mussels, and rodents, and looking at their immune cells to see how radiation and microgravity affect the way that they attack bacteria. So looking at that interaction between immune cells and an invasive organism like bacteria or an infective organism. Micro 5, and Cheryl's here to talk to you more about that, is using C. elegans, this roundworm, as a model for looking at salmonella bacteria causing infection. She'll talk about more about that. There's a JAXA experiment, a Japanese Space Agency experiment called epigenetics, which is also using roundworms or C. elegans. It will look at 4 different generations and their epigenetics. Epigenetics are the outside genetics, the genes that are not in the DNA, but are actually floating around inside the cell. And in looking at those 4 generations of worms, the scientists are hoping to understand whether there's any epigenetic changes in muscle over multiple generations after being in spaceflight. And last but certainly not least, as an example, is Fruit Fly Lab 01, which is a Drosophila or fruit fly investigation, and that is looking at flies with altered immunity and how they respond to microbes. So another way of using a model organism to look at the immune system. There's also a Nanorax experiment on board that has flatworms in it to look at their ability to regenerate, which is something that is often studied in flatworms as a nice, simple model of how tissues and organs can be regenerated from stem cells. So all of the investigations completed their late load either yesterday or today. I think the last late load was complete at 10:52 a.m. And when we do these kinds of life sciences experiments, you can imagine we have to do a lot of lab preparation right here at KSC and then load them at the last minute because they're living creatures or living cells and things. We also freeze a number of our samples and send them up frozen and then thaw them on orbit. Also on the SpaceX 5 flight, we have reflights from some of the payloads that were lost on Orb 3. So 17 of the 18 Nanorax and SESI student payloads, which are also sometimes called Yankee Clipper, are being reflown on this flight, and so that's really important to the students. They're excited about that. And 2 of the 26 Nanorax that were, or Cube Labs that were Planet Labs Dove nanosatellites will also be flying on this flight. And so those are just a couple of the things. I could talk to you about human experiments. I could talk to you about physical sciences experiments and combustion and many of the other things going on. But we really wanted to give you some insight into this cluster of different model organisms that are going up and why those could be important. And I'd like to hand it off to Ken Shields from CASIS to talk a little bit about how model organisms fit in their portfolio and the other experiments going on.
Great, Julie, thank you. And thank you for joining us this afternoon. To start out, I'd like to give you a little bit of brief on CASIS, the Center for the Advancement of Science and Space, and who we are. We are the nonprofit organization that manages the U.S. National Laboratory on the ISS. We do this via cooperative agreement with NASA. We were selected in 2010, began operations in 2011, and SpaceX 5 actually represents the 4th mission, if you will, of projects that CASIS has selected and sponsored for flight on the ISS. Our mission really primarily and very simply, to harken back to a point that Julie made early on in her presentation, our mission is to perform science in space for life on Earth. Just think of it as basically we're out there brokering and promoting investigations to occur on the ISS, that the aims of this investigation or the aims of this project will hopefully benefit somehow life on Earth and also produce some sort of a return on investment to the taxpayer who, after all, paid for this wonderful engineering marvel, the International Space Station. So today I'd like to feature 2 life science investigations that CASIS is flying on SpaceX 5. One is known as planarian flatworms, and planarian flatworms are basically flatworms that have the ability to regenerate organs, if you will, after damage, and also they can regenerate some portions of their nervous system after some sort of damage. This investigation is being flown. It's a partnership by Dr. Mahendra Jain from Kentucky Space and also Dr. Michael Levins from Tufts University. The hope that is the results of this investigation may lead to some improvements in the way that we treat wounds and damage to organs or the nervous system here on Earth. Another investigation that I'd like to highlight for you today is being flown by Dr. Milie Hughes-Fulford from the Northern California Institute of Research and Education, also the San Francisco Veterans Medical Center. Dr. Hughes-Fulford is looking at microRNA expression in space with the hope that it may bring about improvements for pharmacological targets here on Earth. With that, Dr. Hughes-Fulford will do a much better job than I've just done in describing her experience or her experiment in the video that's coming up now. Thank you.
2, 1, 0, and launch of the SpaceX Falcon 9 rocket.
On SpaceX 5, we're launching an experiment from Dr. Milie Hughes-Fulford's lab. And Dr. Hughes-Fulford is going to be looking at the immune system in space and studying how the effects of microgravity on the immune system are similar to the effects that happen in aging adults on Earth.
To dream and to explore is the nature of humankind, and to find new knowledge is key. If you think of it, we all evolved in a gravity field. When we go into space flight and we have microgravity, we have eliminated one variable. In mathematics, if you get rid of a variable, you can solve the equation. And we're able to look at the immune system in a whole new way that has not been possible before.
Dr. Hughes-Fulford is specifically looking at T cells, which are a type of cell that actually orchestrates your whole immune response.
When you have an infection, the T cell turns on molecules that are part of the immune response. What we're looking at are the microRNAs, and the microRNAs are the very earliest molecules responding. And so if you think of the immune system like rain in the mountains going to a stream, going to a river, then ending up in a waterfall down to the ocean, right now you're treating the waterfall. What we want to do is treat the raindrops.
So in microgravity, if we look at the RNAs that are absent from this immune response, we can identify potential therapeutic targets, drug targets, that we can introduce into the cells of aging adults and hopefully promote appropriate and effective immune responses and fight off infection.
I think the International Space Station is an opportunity for the entire scientific world. It's the only place I know that you're not really competing with each other over land or politics. You get together and you work together to make something work.
This project not only involves Dr. Hughes-Fulford's lab here at the VA Medical Center, but it also was sponsored by both the National Institutes of Health and NASA. CASIS can actually facilitate research that brings together multiple parties to really do amazing science and things that are going to really bring back meaningful medical advances to benefit people on Earth.
Thank you. And lastly, I'd just like to also point out that education, and specifically STEM and educational outreach, is a major focus of CASIS. It's really at the center of everything that we do. And I'd like to shine a little spotlight on Dr. Robinson and the fact that she pointed out Student Spaceflight Experiments Program has 17 communities flying on SpaceX 5 that unfortunately were lost in the Orbital 3 launch. And I just want to really pass along my kudos to NASA, to SSEP, and Dr. Jeffrey Goldstein, and also NanoRacks, who were able to turn those communities around and get them on SpaceX 5 in a big hurry. That was quite an accomplishment, and CASIS is a national sponsor of that program, and we really appreciate it. So with that, I guess we'll hand it over now to Dr. Cheryl Nickerson.
Well, good afternoon, and I'm pleased to have the opportunity to provide you with an overview of my lab's Micro 5 experiment, which leverages from my team's previous spaceflight research, which has provided novel insight into microbial pathogens and infectious disease mechanisms. Our previous work has provided novel insight into the mechanisms of infectious disease that could not be observed or could not be found using conventional experimental approaches on Earth, where the force of gravity can actually mask key cellular responses that help us determine whether a cell is behaving normally or transitioning to disease. So for Micro 5, we are going to study the entire host pathogen interaction infection process simultaneously in the microgravity environment. And for our host, our model human host that Julie already referred to, is that little nematode, the worm C. elegans. And C. elegans is a very common model used to study human foodborne infections and how foodborne pathogens cause disease in our intestine, because it has an intestinal tract that's very similar to our own intestinal tract. Our model pathogen is our favorite pathogen in my lab, Salmonella. It's, of course, the leading cause of bacterial foodborne death in the United States. It's the leading cause of bacterial foodborne illness in the United States. And NASA is also, we also don't have any effective prevention in terms of a vaccine or therapeutic for it in the U.S. either. NASA is also very interested in salmonella because it has a potential to get to the ISS. So it's a reason why that food that's destined for the ISS has actually been disqualified from getting there. And it's actually been found in the crew refuse on Shuttle as well. And so if you think about it, our previous work shows that salmonella becomes a better pathogen in flight. It becomes more virulent, better able to cause disease. And you're sending up astronauts, they're immunocompromised up there. That's not a great combination for the crew up there. So there's really three main goals of our Micro 5 experiment, and they are to further our understanding of how intestinal pathogens cause disease in our own guts, to prevent or counteract foodborne illness for the crew, and also to translate these findings from up there back down here on Earth to come up with new strategies to prevent us from getting these foodborne illnesses in the first place, which cost us alone $2 billion annually just from salmonella. So our collaborative Micro 5 team is very multidisciplinary. I'm very proud to represent them from multiple universities, from my lab at the Biodesign Institute at Arizona State University, from the lab of Dr. John Alverdi at the University of Chicago School of Medicine, from Dr. Mark Ott at the NASA Johnson Space Center, from our spaceflight hardware team, BioServe, some of these are going up, at the University of Colorado, of course from our awesome, fantastic ground and spaceflight support teams at NASA Ames and here at the Kennedy Space Center, and also, of course, for the crew who are magnificently going to be doing our science on orbit. So Micro 5 is actually going to be one of the most complex biological experiments that's ever been flown in spaceflight. It's going to be, there's a lot of firsts. So it's going to be the first experiment to ever profile the entire infection process simultaneously in microgravity and follow the entire virulence process, the disease process from beginning to end. It will also be the first experiment to measure the molecular genetic, cellular, and immune response levels, as well as the first experiment to test a countermeasure, a potential nutritional countermeasure, that being phosphate, to see if we can prevent that little nematode from getting sick with salmonella in the first place after it's infected. And just a quick recap, I mentioned this work leverages off of our previous spaceflight research, where we provided new insight into how pathogens like salmonella cause disease in our body. So we previously showed that in multiple experiments, in multiple shuttle missions, salmonella becomes more virulent. So it's better able to cause disease. It's a better pathogen. But that was when the bacteria were flown, okay, only, and they were used immediately to infect animals here at KSC. What was very interesting is that while spaceflight made salmonella a hotter pathogen, the genes that were being turned on and off in salmonella in terms of the virulence genes were not those that we expected when we cultured that organism conventionally on Earth. We further identified a master switch or response regulator that was responsible for controlling salmonella's responses to the spaceflight culture. We went on to show that other microbial pathogens use that same switch, so it's evolutionarily conserved. And that same switch is evolutionarily conserved from bacteria up through humans, and it's found in that little nematode C. elegans. So we're going to be looking for that in our experiment. We also found a way to turn off that increased virulence or hotness that salmonella had on orbit by simply changing the ion or the salt concentrations in which we grew salmonella in spaceflight. And we got a number of clues that might be phosphate, which is the key nutrient to turn off the increased virulence. So that's why we've chosen phosphate as a nutritional countermeasure to see if we can prevent the worms from getting sick when they're infected in spaceflight with this experiment. And then finally, we recently flew a spaceflight experiment where we infected human cells, obviously human intestinal cells, since that's the site of salmonella infection, with salmonella in flight, and we focused on the host response. So we've now seen that the human intestinal cells are responding in really new ways that we don't see on Earth, where the force of gravity can kind of mask some of those responses. So Micro 5 will be the first time where we can bring everything together, because what's been missing is actually infecting a living organism, a living whole animal in flight. So hopefully we can tie those pieces together and try and translate that with new information to help keep everybody, the crew, and us healthy from foodborne illnesses. And I just want to close by just reiterating one thing that Julie said, is that the major advances in our knowledge about biological systems has always come when we study them in extreme environments, whether extreme temperature, extreme pH, and we've always been able to take that new insight and apply it to translational advances to globally advance human health and public life, especially in the biomedical fields. And spaceflight is just the next extreme environment, I believe, that really is going to help offer these new solutions to these types of problems. And so I would thus encourage the public to pay attention to what's going on with ISS science and understand that this is a platform that is already telling us just at this early stage, stage and will continue to tell us a lot about we don't know what we don't know. And if you want to keep up with spaceflight experiments and you want to follow this really cool research on all fronts, we recently partnered with Nature, which is one of the most prestigious journals, science journals in the country, to start a new NPJ microgravity online free access article, which everyone will have access to, no charge, and that will be happening very soon. So thank you very much.
Well, good afternoon. I'm going to tell you about a small experiment that we have that was a result of a competition held by Space Florida for particularly student-developed experiments to fly on the International Space Station. We call it SABLE, or it stands for the Self-Assembly in Biology and the Origin of Life. We got a really talented group of students together and developed a proposal. We proposed to Space Florida, and we're selected, and we've been working on it for a couple of years to get it ready. It turned out to be a little bit more complicated than we thought to begin with, which is not unusual in this business, actually. What we're looking at and trying to understand is a process by which proteins spontaneously assemble from a simple solution of proteins into long linear fibers of proteins. It has interest in a number of different areas. We're interested in it because some of the aspects of when life originated, all of the enzymes that were available that do all of the work to create proteins and form them were not available to begin with. And so whatever happened when we were developing complex structures, it was happening under thermodynamic control. In other words, the temperature and the environment were causing these complex systems to form. We have been studying the growth of protein fibers in the laboratory for a number of years. And if I could have the first slide, please. Ooh, that's the second slide. Go to the first slide. There, thank you. Okay. What you see here is an image of the proteins growing, and it's an atomic force microscope image, and there are two panels there. And the panel on the left is an early stage in the formation of the fibers themselves. You can see they've just formed, the long linear things there are protein fibers. They're essentially one protein in diameter and many thousands and thousands of proteins long. So they're like a couple of nanometers in diameter and as long as 20 microns long. So they form these long, thin, fibrous forms. They grow so the image on the left is an early phase image. It was after about 3 or 4 days the protein fibers were beginning to form. They continue to form for several days, and the image on the right is an image that was taken at about 15 days. And at 15 days, there are so many fibers that have formed, and they settle towards the bottom of the test tube, and they begin to form these fibrous mats. And so fundamentally, we're interested in trying to understand this process, trying to develop an analytical model where we can describe for a given protein how it will form fibers in this manner. The fibers also form complex structures. They wrap around each other and form double fibers and triple fibers. And the internal structure of the fiber changes somewhat. So we're interested in all of that. It also has potential implications in neurodegenerative diseases, particularly if you look at Alzheimer's disease. If you look at the neurons of people who have died from Alzheimer's disease, they find these amorphous plaques inside the cell and outside of the cell. And there are two different proteins. We're using a protein called lysozyme here, which forms rapidly. It's a nice model for us to use. And the other proteins that are associated with neurodegenerative diseases also form these same kind of fibers in a test tube, so in a thermodynamically controlled process. The way we have to do this is in order to start so we want a time series. And we can't do that easily in orbit unless we have something that the astronauts come and take a sample every few days or something like that. So instead what we did is we have 9 separate little test tubes, and if I could have an image of the second image. This is our experiment, and so we have 9 separate vials, which you see the vials, you can see the vial themselves, and they're sort of in a pack as closely as we could get them. And in order to start the incubation process, we have to put the protein powder into a buffer solution, and we have to hold them separate and isolated from each other until we want the incubation process to start. So one of the jobs of the little vials you see there is to inject the protein powder into a buffer solution. And so each vial has two separate chambers. One chamber has the chamber for the solution itself, and that's isolated from another little piston that has the protein powder in it. And when we get ready to start the incubation process, we run a little stepper motor that pushes the piston down into the vial and combines the protein powder with the buffer solution. So the incubation period starts. We also have to raise the temperature to 55 degrees in order for the incubation to occur, for the assembly of the fibers to occur. So each vial also has an individual heater and a heater control circuit. And we raise the temperature to 55 degrees. So they then incubate until we lower the temperature back down to ambient temperature. So by doing this in a sequence, we can cover the growth period starting with a vial that was only incubating for 3 days, one that was only incubating for 6 days, one for 9 days. And so we can cover in roughly 3-day periods. And once the proteins are formed, they're very tough. And so we can bring them back to the laboratory to study them. So we don't have an atomic force microscope on ISS, I don't think, do we? Maybe that's anyway, that's another story. Yeah. So we bring them back to the laboratory, and we have in each individual vial then a sample of the protein growth process in weightlessness, instead of in the laboratory where they will not undergo gravitational settling. So that's the basic experiment itself. We are delighted to get to do that. There were some very, very talented students at Florida Tech that designed and built all of the hardware on this. It was completely designed and built by students at the university. And it's a fairly complex system. And so they designed and built not only the vials, but all of the controls for it. There are 2 circuit boards that have the heater control system. And so it was an excellent project for 3 graduate students and a number of undergraduate students. And I've been really delighted to be part of it and just to try and herd them and keep things going, and pointing out some of the crazy aspects of dealing with fluids and microgravity, which we know a lot about, but it's something that if you don't know about it, you can think fluid floats. When fluid's floating, it's dominated in orbit by surface tension, and if you don't take that into account, your experiment is not going to work. And so it was a really rewarding experience for me, and I'm looking forward to getting it back and seeing what we understand by trying to grow these fibers in weightlessness.
Okay, thank you. We are now ready for questions. We have a number of news media as well as social media gathered here at Kennedy Space Center for the NASA Social. And again, Jason Townsend from NASA headquarters social media is here, and he is monitoring the hashtag #AskNASA. So if you're watching online, please go to hashtag #AskNASA with your questions. If you're on the phone bridge, please hit *1 to get into the question queue. And if you're here in the room, please make sure to wait for the microphone, identify your name and affiliation, and to whom you're addressing your question. And we'll start off with Alan Boyle.
Hi, I'm Alan Boyle with NBC News, and I had a question for Julie, maybe on the big picture of science on the space station and how it was affected by the orbital setback, how you've had to reorganize the program. Looking over this flight, but also over other flights that are to come, how much shifting have you had to do in order to accommodate what orbital is going through?
Yeah, so we have done some shifting, but primarily we've kept the research on schedule per plan and shifted some of the crew resupply around to accommodate it, because a lot of the research is more sensitive to timing. We did have to shuffle some crew supplies around in order to be able to accommodate the reflight of some of the research that we're having now. And it's been, it's really exciting to see the program respond in a situation like this, where now that research is a mission, that assembly is complete, we really do what we have to do around those different systems to make sure the research gets done on schedule and it really serves our researchers. So that's been an interesting thing to watch. That's, I think, very different this year than what you would have seen during assembly when there was a delay in a flight or a problem with a flight.
When you move things from orbital to SpaceX, are there some experiments that have to actually change in order to accommodate a different transport?
Generally, no. SpaceX has, I would say, more late load capability than orbital has, because once orbital stands up, you can't really late load after that. So if it had happened the other way around, we would have had more difficulty reaccommodating from SpaceX to orbital, but going from orbital to SpaceX is pretty easy.
Okay, let's take a question from Jason.
This one goes to Jason Ryan, Spaceflight Insider. This one goes to Sam. And Sam, you talked a bit about the students you're working with, and I was hoping, it's a little bit off topic from the experiment itself, but you've been to orbit twice, and you understand some of the dynamics that are involved there, obviously. So what was it like guiding them and explaining to them how things work on orbit? And can you give us maybe a story or two about what that was like?
Let's see. As I said, fluids behave in kind of they seem to be another state of matter when you're in orbit. And I don't know if any of you have experienced that or seen that, in that they're dominated by surface tension. And for example, if you have a drink container that has a straw on it, they all have straws and they have a clip on them. And that's there for a reason, because if you leave the clip off, the surface tension will pull the fluid up out the straw and down around the outside of the thing, around the outside of the container itself, until it kind of equalizes the surface all over, the water all over the container itself. And so you have to be very careful about designing an experiment, which you normally do in a test tube, because the fluid has a surface on the top and it will sit there, whereas when you build something to use in orbit, you have to take into account the fact that it's going to attach itself to all the surfaces of the fluid itself. And if you have air bubbles in the fluid, the air bubbles themselves will migrate to the center of the fluid, or to the interior part of the fluid itself. And so if you're doing something like we were doing with pushing a piston down into a fluid itself that has a powder in it, there's some complexities involved with that which you don't normally think about, and they didn't think about it, so I had to tell them, describe to them what would happen. Because one of the things that happens is water is an incompressible, more or less, and air is also incompressible, but it will compress somewhat. And so if, in fact, you had a lot of air in the fluid itself, which would happen if you designed it with, a surface like that, when you pushed it down, some of the energy would go into compressing the air, and some of the energy would go into pushing the fluid down. And so, in fact, what you have to do is you have to design it so that you can minimize completely the air bubbles that you have inside in order for this piston injection process to work. And so that was one aspect of that. So.
Okay. Well, I mean, with FIT, with Florida Tech, you guys are obviously, equipped to answer these types of questions. Were there any difficulties that the Institute had in addressing the problems involved with your experiment, or was it something that was fairly easy for you to develop?
No, it was really a design process, like many design processes like this, where you have a scientific objective and you have some, we knew what we had to accomplish. We had to get the powder into the solution and have it mix and raise it to 55 degrees and isolate it from everything else. And we had to do that in a timed way. And so what we did is, had a design team, and there were students from engineering, graduate students from engineering, graduate students from science. And it was sort of an interplay between the science objectives, what are the science requirements and what are the, the engineering requirements to do that. And so it just an iterative process and evolved that way. And I've seen that happen many times in other venues, but doing it with a group of students, it was pretty rewarding, and I think they learned a lot doing it.
Okay, we'll take a question here and then go to Jason for a couple of online questions, and then after that we'll go to the phone bridge and return for questions. Go ahead.
Hi, Leah Cheshire with NASA Social and NASA Georgia Space Grant. I think you mentioned, if I heard correctly, that there were 17 SSEP experiments flying on Dragon, and I was wondering if that included all of the experiments that were lost on the Orbital 3 incident.
So there's one additional SSEP experiment that is not reflying at this time, and hopefully it will be able to refly in the future. And then there are some other experiments that were lost on orbital outside of SSEP. The one that stands out in my mind as probably the hardest to replace, it's going to take the longest to replace, is the Meteor instrument. That was a special camera that was going to look through the WRF, our optical quality window, and get images of incoming meteors as they started brushing the high upper atmosphere. And that one is going to take longer. That's probably the longest to replace, but hopefully it will also get to refly in the future.
Jason, do we have any questions?
We do. We're monitoring the hashtag #AskNASA here. And so this comes from Twitter user Frank, who says, so what is the overall goal of this mission, of the SpaceX launch?
So there isn't a single goal for the space station and for the research that we're doing. There are really 4 goals, and you'll see aspects of each of those goals represented in this mission, in the couple of payloads we've talked about, and in the 250 different experiments going on. So the 4 main goals are to add to our knowledge for future exploration, to have international collaboration and expand our international cooperation to do research, to make discoveries in the areas of life and physical sciences and earth sciences, and then finally to be a foundation for the commercial development of space. So we see commercial partnerships expanding, you see National Institutes of Health getting involved, pharmaceutical companies getting involved, and that is part of expanding that economic development of low Earth orbit, just as you see SpaceX as a commercial vehicle provider and Orbital as a commercial vehicle provider becoming part of that commercial development of low Earth orbit. So all of those things are overall objectives for the space station, and we see those themes running through. You heard Cheryl talk about both the discovery mode as well as applying it back to Earth. So she's trying to discover things about how salmonella infects people, but then she's also trying to work to apply that back on Earth. And you heard Sam talking about the same thing, discovering things about how proteins agglomerate or form strings, and also but bringing that back to Earth and applying it to proteins of interest here. So we see that kind of general theme across all of our life sciences investigations. Our physical sciences investigations, which we didn't really talk about, are more focused on things like fluid physics and combustion. But overall, the big picture is when you take gravity away, you have an extreme environment that defies our intuition. So we don't know what's really going to happen, and that's where the best science happens. And that's really the theme that connects everything together, is we're taking things into microgravity with sophisticated tools, sophisticated genetics, model organisms, special hardware, automated systems. But the end goal is to really challenge ourselves and see if we can get the physics right and get the biology right and learn something new about what's going on.
All right, this question comes from a Twitter user named King here, who's asking what field is the students in who are making this device? If you can elaborate a little on their background.
Okay, that I guess is for me. And the as I said, there were a team of students that helped put the original proposal together, and they were from diverse backgrounds. There were science students, engineering students in general. And the science students were, astronomy students. There was a group called the Students for Exploration and Development in Space that were involved. There was the Physics Society students, and there's a Rocket Society at Florida Tech. And a lot of the students were associated with those, and so there were people interested in rockets and people interested in astronomy and people interested in biology that were all involved. And then the graduate student team that put it together, the primary science student, Dylan Bell, is in astrophysics, studying astrophysics, and I'm an astrophysicist, by the way, so I mean that's sort of a different game for me. And then there were 2 engineering students, and there's one is in mechanical engineering and one is in electrical engineering, graduate students. That's Joseph Sargent and Jose Avendano, and they're the primary team that designed and built the payload. And there were 2 engineering professors, Dan Kirk and Hector Gutierrez, who were also associated with guiding the students on that.
Okay, thank you. Let's go to the phone bridge now where we have a couple of questions, and we'll begin with Frank Mooring from Aviation Week. Frank.
Hi, can y'all hear me?
We hear you fine, Frank.
Thank you. This is for Dr. Robinson, and it harks back a little bit to the previous briefing, if you don't mind. You've talked about using the station as a unique environment. I wonder if you could elaborate a little bit about the orbit of the station and its utility for Earth science as opposed to the more common sun synchronous orbits. And then just a couple of specific follow-ups to that briefing. If you could tell us why HICO is coming down and what's going to replace it, and also any other specific Earth science experiments that you have coming up to fill those empty outside spots. Thank you.
Hi, Frank. Yeah, I'll try to get all of those. That was a lot of questions. First of all, the space station orbit is an inclined orbit, which means it goes around, the space station goes around the Earth but doesn't go over the poles. It goes around the Earth in sort of at an inclined orbit. And it goes around the Earth every 90 minutes, and the orbit precesses, meaning it moves relative to daylight. So the orbits that are usually chosen for Earth science satellites are orbits that cross the equator at the same time every day, say cross the equator every day at 2 p.m. And those instruments get a nice reproducible daily, what we sometimes call climatology, but what they don't do is tell you what it's like the rest of the time. So you can imagine there are certain parts of the Earth that are always cloudy by 2 p.m., especially in the tropics. The clouds start building up in the morning, and by 2 p.m. they're always cloudy. And so you're not going to be able to study the dynamics of the day in those areas. So what ISS orbit, one thing that it offers is the ability to look at places at different times of day and see how things are changing. That also gives us the ability to cross-calibrate instruments. So for example, RapidScat is a scatterometer measuring the winds at the ocean surface, and it will pass under, at certain points in time, every other scatterometer that's operating in other orbits and can take simultaneous measurements. So you'll be able to put all that data together and be sure that all the instruments are taking measurements in a standardized way. Once you know that, you've done that cross-calibration, you can put all the data from all those instruments together and get much more information about the Earth system. So that's just one example of an advantage of the ISS orbit. HICO has had a failure. Its hard drive, basically, if we could access it and put a CD in and reload the software, we would be able to restart it, but that's not something that it was designed to do on orbit, so we can't change it out and service it. So we believe that HICO is at the end of its life now. We have not yet determined what experiment will replace it directly, but there was an experiment coming up that was going to take that site anyway. So right now there'll just be a gap where the site's unoccupied for a little while. Of course, we had intended to operate that instrument as long as possible. Some examples of other instruments that are coming up in the next few years, there will be an IR imager that is helping to understand other aspects of the global climate. There'll be a LIDAR that looks at leaf cover, which is a really important measurement for understanding how dense forests are and what the global carbon budget is. Those are two payloads that were recently selected by the Earth Sciences Division of the Science Mission Directorate. There are also commercial experiments going up. For example, MUSES is a multi-user platform. It's expected to have some kind of an imaging spectrometer on it that will be commercially operated, and that's in partnership with CASIS. And there are several other commercial activities going on for that platform where you've got a sub-platform. You could put up to 4 instruments on it for short periods of time, and the whole platform is pointable. So the MUSES platform will allow that. Also, NanoRacks has a NanoRacks external platform that will provide those same sorts of services for commercial remote sensing development. And I think that answered all the questions.
Julie, just as a quick follow-up to HICO, just to point out, there are thousands of images that are available that are archived via the HICO over the years that can be accessed online. Oregon State University manages that facility. You can go to their website. HICO has a dedicated website. You can also access that through the CASIS website at iss-casis.org.
One more question on the phone bridge before we come back. Elizabeth Howell from space.com. Elizabeth, are you there?
Yes, I am. This question might be for Julie or for somebody else. I was just curious how you plan to contain the fruit flies on the experiment, given that they're just so good at slipping out through those little cracks. Thank you.
Yeah, so our cracks are smaller than fruit flies in the hardware, and so that's basically how they're contained. They're contained. The fruit fly lab is a great system. It has the ability to provide a partial gravity or an artificial gravity by spinning, and it's sealed up tight enough the fruit flies don't get out.
Okay, we're back here at Kennedy Space Center. Go ahead.
Stuart Money, Interspace.net, and my question is for Julie. I wondered if you could talk about going into 2015, the percentage of utilization hours of the station overall. I know NASA sometimes discusses its productivity in those terms. Looking for an update, maybe.
Yeah, so we talk about several different dimensions. You can think of us like a hotel. How well occupied are we? How many of our rooms are full? And we're going into 2015 about 87% occupied, meaning our internal space is 87% full. Honestly, you don't want to be a lot more full than that because then you don't have room to swap things out. So what you're really seeing is the evidence of the demand that we have for all the different researchers that want to use ISS. Another dimension we do talk about is upmass, and because we're reaccommodating some of our upmass this year onto SpaceX, but we're going to feel that pinch until orbital flies again. So that's an important dimension that we're looking at. So we're kind of oversubscribed now for upmass because of until orbital flies again. And then crew time is another dimension that we look at. An experiment like SAM's is completely automated and doesn't use any crew time at all other than plugging the thing in. But other experiments are crew time intensive, especially things like the experiments we're doing looking at the crew's loss of vision and the impacts on their vision. Those take a lot of crew time because you have to have one astronaut operate a tomography instrument and another astronaut gets the measurement taken. So some of those things by definition are going to take a lot of time. The space station was designed for a total of 70 hours a week across both segments, and sometimes you'll hear the number 35 hours a week because that's the US operating segment side of that number. Over the last year, we've been delivering about 45 hours a week, so the crew's been overperforming, and we've been doing that by having the crew defer maintenance on systems where we've learned we don't need to maintain them quite as much as we thought. Engineers are pretty conservative, and so they'll build in a maintenance schedule, and now that we've been operating some of these systems for a while, we realize we don't need to change the filters quite as often, and so we push those systems as hard as we can to get as much crew time as possible. But our crew time is 100% subscribed and oversubscribed, so we always carry hundreds of hours of extra activities on our books so that we're ready to do those if crew time becomes available. So when something happens like the SpaceX 5 slip, it was supposed to launch before Christmas, and then it slipped till after Christmas, and then there were a couple weeks on orbit where we had a variety of activities that could be done that were kind of on hold. They weren't scheduled originally, and we slot those in. And so that's why, for example, Robonaut operated on December 17th, because we could take advantage of that launch slip and because the things that were going to be on SpaceX would have been operated right away and would have been very urgent, and instead that time opened up for other research. So it's always a juggling act. But essentially we're using all the crew time we can spare, and every time we can squeeze the systems and get a little more crew time, there's somebody waiting for it. So it's pretty exciting. The throughput of the lab, as I mentioned, the fact that you've got 100 out of 250 new investigations just in this 6-month period, what you see is scientists are getting up and getting done and moving on. We see scientists flying multiple times, like Cheryl, where she has the first data that she collected. There are even some scientists who disagree with her, have some data they collected. Now she can fly her data and show kind of synoptically what the result is going to be. That is what a dynamic lab is all about. And I've seen the scientists that are working with Arabidopsis, with model plants, arguing about this Arabidopsis experiment versus this one. So we're really seeing those dynamics of a full lab and a fully utilized lab.
Jason is monitoring hashtag Ask NASA, and we'll check back in. Jason?
Wonderful. We have a question here from Twitter user Margaret, who says for Dr. Durrance, once your team knew that Sable would go to the ISS, what was the greatest challenge that your team faced?
Well, the greatest challenge was really the design, and the key component is the vial itself. And I described a little bit about the design process. And we also had to keep the fluid contained. You have to have, with fluids like that, the buffer solution is actually fairly acidic. And so we had some trouble getting through the safety reviews on that, as you might imagine. So we had to design it not only so that it would do what it needs to do, but it would also have 2 levels of containment at all times. So we we came up with a design that within the vial itself, there are always 2 levels of containment for the fluid itself and for the protein, and they're separate tool levels of containment. And when we do the injection process, we remain 2 levels of containment for the fluid itself. So that was really the hardest design process. The other part was it's a little hard, actually, in a university environment like this, to put together a program where you have a program manager, which you would normally have in a large development like this, who's going to manage everything. In a student design payload, you have students sort of coming and going, and so that was another problem that was not really an engineering problem, but it was a problem in sort of getting. There's plenty of interest in it, but students graduated in a couple of years, and so we had to work with a number of different students, and it was the program management part of it was a little difficult in this.
Okay, here in the front row.
I'm Sue Gann Schmidt. I'm with NASA Social, and I'm a children's book author with Chronicle Books. My question for you, Julie, is for our youngest space and science enthusiasts, have you worked with any elementary schools on any experiments? And also I'm wondering if you have any resources or links for them to follow the 250 experiments up there in kid-friendly terms anywhere.
Yeah, that's a great question. We have a couple of things. There's a pilot program that was done through NASA Explorer Schools where students got the opportunity to create summaries of the experiments going on the space station, both and they also did some videos and things, and there was a contest, and the winners of that contest are actually posted on nasa.gov. So when you go to an experiment that one of the students wrote about, you can find a student-produced summary for other students, which is kind of a fun thing. We hope that program will be picked up and continued. It was kind of done as a pilot project. I think it was a great opportunity for students to learn how to take technical information and summarize it, which is something we all struggle with as scientists all the time. But the other thing that I often do, I have a daughter who just grew out of elementary school now. She's in middle school. But in elementary school, when I get taken to an elementary school or go in to speak to students or something like that, I like to use the Toys in Space series. There's a DVD out there, and you can use basic toys, and I'll usually take just 1 or 2 of the teacher activities that are on that DVD, and they can turn into a whole afternoon of activity with the students. So you could talk about friction versus, and does gravity work in certain ways? Can friction counteract gravity? They're just great ways to do intuitive kinds of experiments with students, and then you can show the video of what really happened in space after they've practiced guessing what would happen. What happens when you jump rope in space? And then you can see a picture of astronauts trying to jump rope in space and really think about that. So it's a great way to intuitively start understanding all the things that are associated with this extreme environment. And some of them, even on that International Toys in Space, are really more appropriate for middle school or high school. When you start talking about torques and angles of momentum, that's really high school work at that point. So it's a great resource. I use it all the time.
I'd just like to add for students, high school, junior high students, the new NPJ microgravity journal, it's not just for scientists, it's for kids who have a telescope, kids who are interested in space. It's free online access for them to learn and follow what's going on with microgravity research. That's also another great free online resource for them.
Go ahead.
Hi, Val Phillips, Zero G News and Florida Tech Crimson. It's for Cheryl and Sam about the student involvement that's been in respect to these experiments. Particularly with Sam, it seems to be very interdisciplinary. How has it been to manage all the various departments you're involved in? And Cheryl, I'm not too sure how wide range your student body is. How have you both been mentoring students to come forward and hopefully progress further into doing microgravity experiments?
Sam, you start, I'll go after you.
Okay. As you said, it is a very interdisciplinary project itself. And so it was a fairly interesting process because there are 2 colleges. There's the College of Science and the College of Engineering, and they have different management structure. And so we had to put together a way that we could use the budget that we had effectively in both the 2 colleges And so that you kind of asked me about the management aspect of it. It worked out fine. I mean, everybody was all very enthusiastic about it. The students and the all the way up to the president of the university. And so, but there were some difficulties in trying to manage and keep track of where we needed the resources and what resources needed to be done. And so we tried to set up regular meetings so that we could go through this interactive process. And it's fairly difficult with a group of students because you've got classes, you've got exams coming and things like that. And so it worked out fine, but it was interesting process.
Yes, in our case, it's very multidisciplinary as well. We have students flocking to our lab from physics, engineering, and life sciences. Our forte is in biological sciences and microbiology and immunology and infectious diseases, but we've worked with computational undergraduate student mathematical science majors. I'm really excited because we always have a really big waiting line at ASU to get into our lab. We have about a 2-year waiting line to get into the lab. And so every one of the undergraduates, especially my team members here in the back, Jennifer Barilla, Richard Davis, Rebecca Forsyth, and Ji-Sun Yang, they've been here for 2 months now with this experiment. You can imagine with undergraduates, they can't be gone for 2 months. And so they get to be involved with a lot of our translational work where we use ground-based spaceflight analogs to try and recapitulate what we see in flight so that we can refly again. It's a huge team effort between our institute and scientists and students at all the other of our collaborators' institutes. They get to be online with us. They have bi-weekly telecons where they have to present their results to our NASA collaborators, to our University of Chicago. I mean, they're all there and they're so jazzed to be a part of it. And it's great because you're inspiring. I think all of us see so much fun in being able to inspire the next generation of scientists who are really going to take this platform to the next level. And just one quick thing I want to follow up with Julie said. It's very important to have intellectual debate and sparring. That's what scientists do. We love to do that. But and challenging each other's findings experiment is great, but you've got to make sure that's being done by people and scientists that are using hypothesis-driven, well-planned, well-executed experiments. There's no excuse for any experiment not being done that way, especially in the unique environment of spaceflight. And the group you're referring to, Julie, just didn't have well-planned experiments to be able to challenge. Their results were completely invalid. So that's all I want to say. Thanks.
Okay, we have time for 2 more questions. Right here.
Hello, Tara Foster from NASA Social and TaraStrellis.com. I'm just wondering, what's the success rate for your experiments returning to Earth? Because it's kind of not like driving a car around the block.
They all return on SpaceX, and so far we've returned all of them. So we're doing pretty good on that. SpaceX is a good provider for us.
Yeah, we're certainly anticipating getting them back because we don't get our data until we get them back.
Right here.
Hello, Laurel Ann Whitlock with NASA Social, but my question actually comes from my Twitter friends list, at Doc Foreman on Twitter. She wants to know what is being done outreach-wise and engagement-wise to help citizen scientists, especially those very active on social media platforms such as Twitter or Reddit, to become involved in this, to become engaged in this, maybe to watch the experiment, maybe even contribute in some ways.
So there isn't a single citizen science approach. There's no centralized citizen science, just like there's no centralized anything on the web anyway. But there are a number of different ways to follow the work. At ISS_research is my Twitter tag, and you can follow that. And we have a variety of reports on what's going on every day. Things that are happening. We retweet a lot of our scientists that tweet, so you can hear their perspectives about what's going on. So that's a really great place to watch if you want to see kind of the day by day. There are also specific experiments that do have online components. One that jumps to mind is ISS Biomed Online, which has a number of different experiments that have flown in the past, and they're set up in a way so that teachers can still stream them and use them in the classroom even though they're not live, because there's one set of students who are all excited about, say, spiders in space in one period of time, but 2 years later the students never heard about it. And so teachers are actually able to use that, recycle it, and only let their students see the amount of data they should see and use that curriculum year by year and over and over. So that's another thing that happens in the online universe that I think is pretty exciting.
As a follow-on to that, CASIS, we actually have via our website a portal where citizen scientists, commercial companies, anyone with a good idea can submit what we call an unsolicited proposal, which in a way is a non-traditional way to access the ISS and utilize the ISS. Typically, access to the ISS has been through research announcements via NASA or some other large organization, NIH or others. However, we're open to great ideas to fly research 24/7, 365. You just go to iss-casis.org, go into the Opportunities tab, and the instructions are right there. Very straightforward.
Okay, we're going to close the briefing with some closing remarks from Julie.
Great. Well, I think you've heard today a couple of themes I just wanted to pull out as we close. First of all, I think the theme that our intuition is really not sufficient to guess what's going to happen when we take gravity out of the equation, and that is the perfect extreme environment for making scientific discoveries. And we're using that extreme environment to challenge biological systems and learn how those systems are working. But we're not just doing it to make a discovery, but also because that knowledge comes back here on Earth and can be applied to very real problems like foodborne illness, problems like other diseases, problems like muscle loss. And so you're really seeing the complete picture of how research in a platform where otherwise you could never learn this stuff can make such a difference back here on Earth to our lives. And so it's amazing to see an active, dynamic, full laboratory, what that really looks like. And you've just seen a couple of folks here gotten to drill into their lives and the exciting work that they're doing, and just to realize that there are hundreds of scientists out there with these same kinds of stories to tell. The space station is really an amazing laboratory. Thanks.
And to find out more about the research and technology and other activities surrounding the International Space Station, another website to go to is www.nasa.gov/station. And that will conclude this briefing. Our next briefing will be at 4 p.m. Eastern Time. It's the SpaceX CRS-5 pre-launch news conference, and we will see you then. Thank you.